Okay, we are going to look at the database question today for cellular respiration. Here you can see the setup is given where the yeast is in the solution. There is yeast, sugar and nutrients, and the setup is put on the weighing scale electronic balance, which is attached to a data logger. So we are recording the change in the mass over the period of time. Here, to prevent entry of oxygen, airlock is done. So it is preventing the entry of oxygen. So it means it is anaerobic respiration happening. First question is calculate the total loss of mass during the experiment and the mean daily mass. So there are two parts of this question. The total loss of mass, so you can see they're starting from 560 and the mass after 10 days 545. So total loss of mass, this is the first question. Mass, you can write down 560 minus 544 grams. So it's 16 grams. Second part is daily loss. So it's for 13 days, as it is for 13 days, 16 by 13, around there will be uncertainty per day. It's probably 0.1, I'm not sure. Here uncertainty can be accepted. Second question explain the loss of mass. So why there's a loss of mass in the solution? This setup is having a loss of mass, you have to explain. So as we mentioned, it's how many marks question? This is a 2-mark question. Again, sorry, this is a 3-mark question, so we need to have 3 marks. So first mark should come, you have to identify anaerobic respiration, cellular respiration. You can also call it alcoholic fermentation because it is yeast and anaerobic. So CO2 is produced, which is the waste product as we know from anaerobic cellular respiration, and release of CO2 leads to loss of mass. So that should be 3 points for this question. Next question, 2 reasons for the increasing rate. So from here to here you see the increasing rate of loss of mass from the start until day 6. Until here, day 6 to 7, you can see increasing rate. So what is the reason? 2 reasons they're asking. So their nutrients are there. It is yeast, sugar solution. So definitely there's a population growth of yeast. And what exactly is happening here? More yeast leads to more respiration or high rate of respiration. So it is an increasing amount of CO2 produced or concentration of CO2 produced. So these can be 3 points. I think that is good enough for 3 points. You can talk about you can go in detail, like waste heat is also produced, which is decreasing CO2 solubility. But I think these 3 points are good enough to get the marks. Sorry, this should be question number 3. Let's look at question number 4. So this should be 3 points here. We need to write down only 2 reasons, so any 2 reasons for there. Now suggest 2 reasons for the mass remaining constant from day 11 onwards. So this is constant. So why is it constant? We can see the substrate. What is here happening? The substrate becomes the limiting factor. And then another thing you can see, the yeast, because there's a high alcohol, can lead to high alcohol leads to death of yeast, and that can cause yeast, that can also cause the decrease. So two reasons that should be good enough to get you full marks.